Thank you for joining us. We're very proud to host tonight's lecture by Dr. Ruth Feldman. She's an internationally recognized expert in parent-child relationships. She has degrees in music composition, neuroscience, clinical psychology, developmental psychology, and psychopathology. So her approach integrates perspectives from neuroscience, human development, philosophy, clinical practice, and the arts within an interpersonal frame and a behavior-based approach. Her conceptual model on biobehavioral synchrony systematically describes how a lived experience with close relationships builds brain, creates relationships, confers resilience, and promotes creativity. Her studies were among the first to detail the role of oxytocin, the love hormone, in the formation of human social bonds. Her research is translational and informs the development of various interventions applied around the world. Her tools are used in 17 countries and translated to multiple languages and utilized in research on all facets of human relationship. She's a consultant on multiple international grants and a frequent keynote speaker in international conferences. Her studies have followed children from infancy to adulthood, and we'll have a chance to learn about that tonight. With over 300 published research articles, she's one of the world's most cited researchers. She's a fellow of the Association for Psychological Science and received multiple awards, including a Young Musician Award, Rothschild Award, NARSAD Independent Investigator Award, and the Gravens Award for research on high-risk infants. So please join me in welcoming Dr. Ruth Feldman.
Okay, good evening. It's a pleasure to be here. I'm going to talk about the neurobiology of love, synchrony, and the human affiliative brain in health and psychopathology. And I thought I'd start by some images. Louise Bourgeois, an artist, a feminist, a mother, a writer, who throughout her career was fascinated with mother's body and the birthing process. The wonder that we come out of someone else's body and what profound implication this has on us, on our brain and behavior throughout life. What does it mean to be born a mammal? Mammalian young are born with two important constraints. Their brain is immature at birth, and young maintain close proximity to a nursing mother. This means that the mother's mature brain externally regulates the infant's immature brain in a system-by-system way and tunes it to social life through a process we call biobehavioral synchrony. And what is biobehavioral synchrony? It is defined as the coordination of biology and behavior between mother and young, between mother and infant, during moments of social contact. We found that when mother and infant, beginning in the 3rd months of life, coordinate their nonverbal behavior in the gaze, affect, vocal, and touch modality, these coordinated, precious social moments provide a template for the coordination of physiological processes. So during those moments of joint action, of synchronized behavior, there's a coordination of mother and infant's heart rhythms, which is not found during non-synchronous moments. There is a coordinated release of oxytocin, which is not found after non-synchronous interaction. And there is, during these moments of coordination, of social coordination, brain-to-brain synchrony in key nodes of the social brain. This is the way by which synchronous behavior coordinate mother's and infant physiology, and this means that we're taking a behavioral-based approach to the neurobiology of love. This experience of synchrony between mother and child during the sensitive period of birth to 9 months, before the brain reorganizes to language, have two important implications. One, along the attachment relationship. So like expanding ripples, these rhythms that were set during the nonverbal stage incorporate into them the child's newly emerging social ability. Mother and toddler play in joint symbolic play, In preschool, there is the co-construction of imaginary scenario. When the child becomes an adolescent, there is the beginning of empathy and the ability to see the mother's perspective until the mother and child can engage in a full adult-to-adult relationship of mutuality, intimacy, and perspective taking. But this is built on the same rhythms of the synchronous early nonverbal dialogue. And the second important implication of this synchronous experience during the sensitive period is across human relationships. So if we have behavioral synchrony of nonverbal signals, when we look at each other, when we smile together, when we co-vocalize, when we affectionately touch each other. We have autonomic synchrony of heart rate, we have hormonal synchrony of oxytocin, and we have brain-to-brain synchrony. These are found in various forms across human social relationships, between parents and infants, both mother and father, between romantic partners, among close friends, and also what we call fellow humans, strangers. Humans are the only species that can activate their affiliative biology to the pain or joy of strangers, either stranger we meet on the streets or even those we see on televisions. Our ability to engage in other relationships with partners, with friends, with strangers, and eventually parent the next generation are all set by the experience of synchrony we experience during the first months of life. The neurobiological system that supports all of it is the oxytocin system. Now, the oxytocin system is a very ancient system. It's presumably evolved approximately 500 million years ago via gene duplication in jowed fish, and it's found in all vertebrate and some invertebrate species. The role of the oxytocin molecule in these ancient species was to help organisms manage life in harsh ecologies. So oxytocin was implicated in water conservation, in thermoregulation, in energy balance. With the evolution of mammals, oxytocin became intimately and causally involved in the mammalian condition by controlling birth and lactation. What happened then is something that we should take note of because it is critical to all the work on disrupted bonding that we do. Mammals learned to manage hardship through relationships. So they no longer learn to manage hardship in a group like fish or ants or birds. And the mother-infant bond became the context where you learn to manage hardship and you develop the system that can sustain stress regulation. But this, what it did, and since humans are the primate species that requires the most maternal care and the longest periods of dependence, is that those systems, the oxytocin system that support love and bonding and empathic behavior and consoling behavior and group sharing also became intensely sensitive to danger, intensely sensitive to stress. And this is why love can turn very quickly into fear. And this is why oxytocin that sustains bonds and stress management can also sustain group derogation, parochial thinking, prejudice. It's us versus them. So here is where we need to be very careful, where the neurobiology of love can very easily turn into the neurobiology of fear. Now, my lab has been working on oxytocin for over 15 years. I think we've published hundreds of papers on oxytocin, and we have managed to assay oxytocin from nearly every fraction of the human body, from blood, from saliva, from urine, from hair, from sweat, from breast milk. So I wanted to just give you one slide of a quick summary on all our work. It's impossible, but just something, an overview on our work on the involvement of oxytocin in bond formation. The first work we did about 10 years ago and it was very surprising at the time, is that mothers and fathers have similar levels of oxytocin when oxytocin is not measured just before or just after breastfeeding. And this was surprising because oxytocin is the woman's hormone. It's implicated in, it rises during birth, and it's triggered during oxytocin causes the contraction of the uterus, and it causes milk letdown. So you'd think that this is a feminine hormone, but no, it seems that fathers are as prepared as mothers to care for children, and I will show you that when we talk about the paternal brain. So this was a very not common finding or surprising finding. It was contrary to our hypothesis, but since then, us and other labs found it in hundreds of fathers. So fathers with work in a different pathway can release as much oxytocin and can support positive parent-child interaction. Second thing we found in several studies is that oxytocin is released following touch, contact, and synchronous interaction with close others. So between parents and infants, or between romantic partners, when you touch, when you keep physical closeness, or even when you engage in synchronous interaction, there is a release in oxytocin. Third thing is that we found in loving partners, when you fall in love. We studied partner during the first 3 months of falling in love, and what we found is that the highest levels of oxytocin in a human's life is during the period you fall in love. It's even higher. It's about 2 times higher than when you become a parent. So you all remember the feeling that you feel like you're walking on cloud 9. You're probably walking on very, very high levels of oxytocin. And for those who remain in a close relationship, a year later, oxytocin levels do not go down, okay? So when you're in a good, loving relationship, those maintain oxytocin at high level. And the last thing is that when bonding is disrupted, the first system to be disrupted is the oxytocin system, because the oxytocin system is the most epigenetic of all neurobiological systems. It is highly dependent on caregiving. So when bonding fails, conditions such as postpartum depression, high stress, poverty, premature birth, all conditions that do not enable a smooth transition into maternal-infant bonding, they would disrupt the oxytocin system. So let me just walk you through how I'm going to arrange the talk tonight. The first thing I would like to describe to you is the parental brain, the maternal brain, the paternal brain, and what is the role, the system, the neural network that comprise the human parental brain. The human parental brain is also the same neural network that underpin love, friendship, and then our capacity to parent the next generation. The next part will show the long-term implication of the parental brain to show that it's not just neural networks that coalesce and work for the time of infancy, but that they have measurable and important consequences for the child's social and emotional development. The third part will ask the question of how relationship become brain. How do the child, out of this bond with the mother and the oxytocin that was built, builds the social brain? The social brain is an environmental dependent brain structure. And the last part, I'd like to talk about translation to intervention, how synchrony, I would like to describe to you a synchrony-based intervention for postpartum depression. So let's take our trip into the maternal brain. The research on the maternal brain began approximately 70 years ago in the early 50s with the work of J. Rosenblatt and colleagues who wanted to understand the brain structures and brain networks that enable rodent mothers to care for their infants. And they found that there was a central role for the MPOA. It's a structure in the hypothalamus. The hypothalamus is the brain's oxytocin factory. The hypothalamus projects to the VTA, the dopamine factory, to make the infant the most rewarding stimulus for the mother. It also projects to the amygdala to increase maternal vigilance. There are similar network working in fathers, and recent work shows that it's the same brain structures but different neuronal population implicated in mothering and fathering. And this comprises the mammalian maternal brain that's found in all mammals from rodents to elephants and enable mothers to care, to invest, to create a secure habitat for their infants. Now, what is important here is that just want to show you that these mammalian brain, they did not look at cortical areas, and they did not look for functions that are higher than the subcortical automatic. Now, if you look at the human brain, what you see here in C, this is the subcortical mammalian maternal network. And what you see in A and B is that this mammalian network implanted into the human parental brain. So what you see here is that the subcortical structures that enable us to love are really copied from these evolutionary ancient mammalian maternal brain, that those give immediacy and reward and the automatic fear we have for our infants. And it's subconscious, it's immediate, and it's uncontrolled. Maybe this is what Freud meant when he said libido, something that gives our love a sense of urgency and a fire. But on top of it, you see large cortical areas that are connected via multiple ascending and descending projection to those subcortical structure. And in its totality, they create the human parental brain. This is a work that we did summarizing all papers that up to 2015. There's several more from that in the last 5 years on the human parental brain. And they showed that in the human parental brain, there is the subcortical network just like in other mammals. But these are human-specific network that are critical to parenting and are implicated on top of them. The empathy network that enable parents to feel the infant's pain and state and emotion in real time. The mirror network, the empathy and the mirror network together called the embodied simulation network, the mentalizing network that enable the parents to give meaning to the child's nonverbal communication, and the latest evolving emotion regulation network that enable parents to multitask, to plan parenting according to long-term goals, and all those systems together comprise the human parental brain. So we have this subcortical network that we share with other mammals, and on top of them, network that enable us to feel the infant's emotion, to mentalize what the infant intends to tell us, and to plan our parenting according to our cultural meaning system and what we feel is good parenting. Now I'd like to show you some studies we did on the human parental brain and to see the role of synchrony in the human parental brain. And the way these fMRI studies, these are all fMRI studies, and the way they work is because the key component in human attachment is the exclusivity of it. The fact that I love you means that I don't love the person to your left and not the person to your right. I love you specifically. And the enduring component of attachment, when I love you for breakfast, lunch, and dinner, and I don't change you every time. We show parents videos of their own infants, and they compared with videos of unfamiliar infant that is matched for age and for race and etc. And we collect parent-infant interaction in the home ecology. And this is where we follow the work of Lawrence, if you remember, with a duckling, that he said that in order to understand bonding and the role of bonding in development, you have to go to the pond and look at attachment in its natural habitat. So we go to the home, we videotape parent-infant interaction, and then we take those interaction and we tailor-made the stimuli in the magnet, and parents see their own interaction versus unfamiliar interaction, and we see what is being activated by your own interaction. So this is the paradigm. So in the first study, we wanted to see how synchrony is implemented in the maternal brain. So we collected 45 mothers, and we divided them, we microcoded the interaction, and we divided them to two types: synchronous mothers, who engage in interaction that is matched to the infant state, that is responsive, that engages joint looking and joint smiling, and the infant feels relaxed and loved, versus intrusive interaction, where mother overstimulates the child, is not waiting for the child's signals, doesn't, and often involves a physical manipulation of the infant. And we wanted to see how those mothers engage. The first thing we saw when mothers, those two types of mothers, observed their own and unfamiliar interaction, that there were very different levels of activation in two brain areas. One in the amygdala, which was much higher in the intrusive mothers, and one in the nucleus accumbens. It's a key area in the dopamine reward system of the synchronous mothers. And that tells us that synchronous mothers, the synchronous parenting is underpinned by joy, by reward, by mothers feeling that they are attracted to the infant. Whereas intrusive parenting, we often see that with stressed or depressed mothers, is underpinned by anxiety and fear and a sense of an amygdala vigilance. The next thing we did was to take these two seed regions, the amygdala, the fear, and the nucleus accumbens, the reward, and say what synchronizes with those activations throughout the observation. So the mother was watching 40 minutes of videos, herself with the infant, somebody else with the infant alone, the mother alone, etc. There was a whole 40 minutes of videos. And we wanted to see what synchronized in the brain with these two seed nodes. And what we found is that for the synchronous mothers, together with the nucleus accumbens, the reward coloring, there was all these other networks that were activated. The whole parental brain was working in concert. Whereas the amygdala, there was nothing really working with the maternal brain. So that means that when the underlying coloring of parenting is that of reward, is that of approach, there is an integration into a synchronous activation of this parental brain. But amygdala activation, this over-fearful activation, does not allow the mother to empathize, to mirror, or to mentalize to see what the infant really needs in real time, does not allow the mother to be present fully with the child. In the next study, we wanted to see what's happening with the father's brain. And we wanted to see what happens with father's brain in relation to two things. We wanted to see whether father's brain activates like mother's brain, and whether the differences between the maternal and the paternal brain relate to biology. The father's brain was not primed by oxytocin during pregnancy, birth, and nursing, or it's because of the primary caregiving role. Because in most countries, even in the most advanced country, mothers are still the primary caregiver. Think about it. Most of the time, young kids in kindergarten, the kid is sick, they will usually call the mother. It's usually the mother who is the primary caregiver of the child. So in order to study that, what we did was recruit nearly 100 families that comprised three groups. We had mothers and fathers who were heterosexual couples, and we had 48 homosexual couples that were living in a committed relationship, because single parenting, it's a whole different story in the parental brain. And they had a child through surrogacy. So they had the child since birth. In each gay couple, one father was the biological father, and one father was the adoptive father. But they both considered themselves primary caregiver. We had long questionnaires and interview about who is the primary caregiver, and they both differed from the secondary caregiver of the heterosexual fathers. And then we exposed them to the same paradigm. We showed them in the magnet. We went to visit them. We videotaped each parent. We videotaped the infant with each parent. We videotaped the infant with both parents. And then we exposed parents to these videotapes in the magnet in relation, in contrast with unfamiliar infants. Again, mother with mother, father and father. And what we found is that this parental brain is a very, very widespread, okay, widespread, so across all parent type. Mothers and fathers, biological parent and adoptive parents, primary caregiver, secondary caregiver. When you become a parent and assume the role of taking care of committed care for this child, your parental brain will activate. So you see all the subcortical mammalian network activated in every parent, the embodied simulation network, the empathy in the mirror, and the mentalizing network. And it basically activated to the same extent across parent. And I like to think of it like that until 150 years ago, every third mother died at childbirth. Who was the ones that raised children? These were neighbors. Women who did not give birth right now took care of infants, and infants generally survived. And that means that when you take upon yourself the role of a parent and you're involved in infant care, this bottom-up brain network organization would trigger the activation of your parental brain. However, there were two areas that show difference between mothers and fathers. The amygdala activated in mothers 4 times more than in fathers, and the STS is a key node of the mentalizing network. Okay, activated about 3.5 more times in fathers than in mothers. And we like to look at it as that there are 2 pathways to parenting. The maternal pathway, which is evolutionary ancient, it's automatic, it's subconscious, and the paternal way, which is more secondary, cortical, and controlled. And it comes through actively caring for, being with a child and actively caring for the child and trying to imagine what are the intention of that child, and that these are 2 different pathways to activate the parental brain. But what you see in the light green is really surprising. The gay fathers, the primary caregiving fathers, had amygdala activation just like mother, and STS activation just like fathers. And the question is, what is the mechanism that makes these amygdala in these men activate 4 times more than in other men? How could they get their amygdala so much without the hormones of pregnancy and childbirth? Okay, so we were looking for mechanism that made this amygdala activate so much. We were looking for correlation with synchronous parenting and with oxytocin, and this wasn't the story. In fathers, both types of fathers, it correlated with the STS, and with mothers, it correlated with the amygdala or areas of a paralimbic system. And then what we found is that only in the primary caregiving fathers, the amygdala activation synchronized with the STS activation, and the 2 areas work together to keep infants from harm when there are no mothers present. So think about it like this. Every mother knows it. You go to sleep. You have a 15-year-old. She went to a party, okay? You know who she's with. She's in good company. You arrange for your best friend to pick her up, so you know she's going to come home. You go to sleep, but your amygdala is open. It's 3 a.m. in the morning. You hear the door open and her footstep tiptoeing in. You turn to the other side, and you begin to sleep in earnest. I believe that mothers, from the time an infant is born, mothers sleep with their amygdala open. And I'm telling it to you, and my daughters are already old. And fathers sleep with their amygdala closed. And we know it from very... And then, comes the middle of the night, and he's a good father. The baby needs a feed. The baby needs... So the father will wake up, and will feed, and will diaper, and will take care, and will burp, and will do all the things, and will go back to sleep, and his amygdala will turn off. Now, an infant needs someone who will in his or her amygdala always. Without someone keeping the fragile baby in mind, that fragile baby may not survive. So what happened when that child only has 2 dads? Someone's amygdala's got to be open all night. So this is what happens to the very plastic parental brain. The STS, which is the paternal brain, recruits the amygdala, and the two work in synchrony, making a new neural network of the maternal way and the paternal way, so that there is somebody who can watch a child when there is no mother to take care of him. By the way, we are following these kids. These kids are now 8. And we see differences between children raised by good parents and by less good parents. But we don't see any differences. And we look high and low between children raised by 2 good fathers versus those raised by a good mother and a good father. What are the long-term implications of this parental brain? Are they all nice colors that you see on the board, or do they have some implications so that when there is greater activation of the parental brain, there's some benefit to the child's later development? So we checked for emotion regulation and socialization when children were about between 3 and 4. We checked for socialization during a typical pickup task, regulation of negative emotion with a mask paradigm. You microcode both how much negativity the child expressed and what was the ways the child regulated his or her fear. And we checked for regulation of positive emotion with a soap bubble play paradigm, whether the child expressed positive emotion and what was the regulation strategies they used to regulate their emotion. And we used a network coherence indices, and this is a complicated mathematical way to see how structures in a network combine together, work together as a network. So the subcortical network, how well it worked together, the embodied simulation, the empathy network, how that worked together, and the mentalizing network, how well it worked together. And what we found is that children whose parents 4 years prior had greater cohesion of the subcortical system expressed more positive affect and used more simple regulatory strategies to manage, like proximity to the parents. Children who had a greater regulation of the embodied simulation network were able to express less negative emotion and used complex regulatory strategies like talking to themselves, gaze averting, or using symbolic plays. And children who had parents whose mentalizing network was more cohesive were more socialized. Moreover, we followed children until 7 years. So here you see that parents' embodied simulation network predicted the child's complex regulatory strategies, but only children who received synchronous parenting in infancy. This same system, empathy network, predicted less child internalizing disorders at 7 years, pending the child's ability to use regulatory behavior at preschool. And the activity of the core limbic and its connectivity with the empathy network predicted the child's oxytocin when the child was 4, pending the parent had higher level of oxytocin. So here you see that activation of the parental brain in infancy predict the child's socialization, emotion regulation, and mental health, but pending that child received synchronous parenting and the parent had high oxytocin levels. This, I'm going to skip this so that I have time to go to this. And this is the new line of work in my lab, and this is brain-to-brain synchrony. This is hyperscanning techniques, and that you see the parent and the experimenter and the infant both wearing an EEG cap. And what you measure here is the synchrony between the brain activation and not exactly how much the brain activation, we're not looking at how much there was a brain activation, but how the two brains synchronize during social activity. And what I want to show you, just preliminary finding from three types of interaction so that you see the synchrony in brain waves. This is the baseline. They have a screen between them. They don't see each other, and each one is looking at a different computer. So the baby is seeing soap bubbles or something, and the mother is seeing something else. So they're not synchronized because of the same stimulus. And here you see the waves are not very synchronous. Here you see the free interaction, and you see that the waves begin to synchronize. And here you see mother and infant during breastfeeding, and you see so that breastfeeding and this physical proximity and touch to a very close match, a very synchronous an infant's brain. This is in the theta waves. Theta is the most predominant brain wave of the infant, of the maturing brain. This is in the gamma waves, and you see the same story. No synchrony during baseline, more synchrony during face-to-face interaction, and perfect synchrony during breastfeeding. What happens when there are disruptions to maternal-infant bonding? I've described to you the 3 legs of the neurobiology of affiliation: the parental brain, the oxytocin system, and biobehavioral synchrony, and that these 3 processes work in concert but are also very, very sensitive to disruptions. So there are 3 longitudinal cohorts. There are 3 types of disruption to the neurobiology of affiliation, and we follow each cohort like that, like these 3 disruptions from birth until adolescence or young adulthood. And today I chose to tell you about the postpartum depression. So one very common type of disruption relates to early life stress and trauma. It disrupts the mother's ability to focus on the infant's need. And I think this disruption to the mother's attention and ability to be present is even before the disruption to whether the mother can physically provide for the infant need. Because even if mother could provide for the child's basic need, most of the time this chaotic living and fearful and trauma doesn't allow the mother to really be peaceful and to be present and attend to the infant need. The second is postpartum depression. We're talking about 18% of women in industrialized society, and the numbers really go higher every 5 years. At 2012, it was only 12%. At 2017, it was already 18%. So we're talking about millions of mothers each year. And the third type of disruption is because of physical separation between mother and infant when infants are born prematurely. So those infants whose brain is the most premature and require the most proximity to the mother's body actually get the least of it. So we're talking about really million here. Every You know, you have millions of children that are living in poverty and chaotic situation. There's between 10 and 11% of the babies born in Western societies are born prematurely, and 18% of the mothers. So we really are talking about staggering numbers when bonding fails. So when all these pieces, complex pieces, do not fall into a puzzle, there is a disruption, and disruption during this period will carry long-term effect on the children, and they will transmit it to their children. So here I'd like to describe to you birth to adolescence study of maternal postpartum depression. I'll walk you through what we did and just pick a few findings from the 15 years of follow-up. And I thought I would start with two pictures from Juan Miró. Juan Miró was an artist, a famous Spanish artist. He suffered from depression, from major depression, bouts of depression throughout his life. And apparently he was also a child of a depressed mother. It's possible that she suicided at some point in his adolescence. So I thought these are two very interesting example. Head of a woman, here the child describes his internal object, what he feels the mother is, and you see a mother's face as disjoint and even ominous. And here he describes a portrait of the self as impoverished and barren. And the question is how you get from the picture on your right to the picture of your left, when the mother is not experienced as welcoming and containing, and there are lifelong disruptions to the self. So this is our study, and we modeled this study as close as we could on Michael Meaney's studies of rats. If you remember, Michael Meaney wanted to see the effect of maternal licking and grooming, good caregiving, sensitive parenting on the infant brain. So what he did, he took a bunch of rat mothers, and he watched how much they licked and groomed. There is natural variation. Some did a lot of it, some did a little of it. He took the two ends, the high lickers and the low lickers, and he bred them for several generations. And then he had the good moms, if we call them this way, and the not so good moms. And then he saw what happened to the child's brain. We thought of postpartum depression as representing the same situation, and we took an ecological approach to studying it. And this is how we did it. We first collected 1983. Everybody makes mistake. It's not a year. It's a number. It's close to 2,000 mothers. And we recruited mothers who are normative from the population. Remember, there's about 15% of mothers who would go on to develop postpartum depression. And in order to tease depression, we wanted to take low-risk mothers to really see what the effect of depression per se, not when it's combined with poverty and single motherhood and no father and et cetera, et cetera, et cetera. So we took only mothers who were above 21 years, above the poverty line, married or cohabitating, and that at least 12 years of education, so the family was of low risk, and we controlled only for depression. Not only that, about a third of the depressed mothers are comorbid between anxiety and depression, so they are depressed and anxious. Depression lowers oxytocin production. Anxiety increases it. So we wanted to study only the depressed mothers to see what depression does to the child's brain. So we also excluded mothers who were anxious. So we started with mothers in the hospitals. When they gave birth, we went into them on the second post-birth day. We asked mothers to complete questionnaires, and we recruited them to the study. And then we took those who had the lowest depression and those who had the highest depression, and we excluded cases of anxiety, and we turned to 680 mothers when the children were 6 months old. And then again, we took the lowest depression group, the highest depression group. We turned to 254 mothers when the infants were 9 months old, again excluding mothers in depression. Then we had at 9 months 192 mothers that we visited at home. We had a home visit. We did a clinical diagnosis, interaction. We collected homework. We watched for the child's emotion regulation. And here we have mothers who are clinically depressed. So our mothers who are clinically depressed at 9 months are those who reported high depression at birth, 6 months and 9 months, and were diagnosed at 9 months. So they were depressed throughout the first year of the infant's life. We then followed them at 6 years. We went to 156, whoever we were able to track and was willing to take us. And over there, we saw mothers and fathers. We collected hormones from mothers and fathers. We saw observation, and we diagnosed mothers and fathers and children. Okay? And then we formed 2 groups. We had 103 mothers who were healthy, reported no depression, no anxiety at any time point, were not diagnosed with depression, and they were not diagnosed with any other disorder. So these are our healthy mothers. And we had 46 depressed mothers who were depressed throughout the child's first year, received a clinical diagnosis at both 9 months and 6 years, and reported being fairly depressed throughout the child's first 6 years of life. So we have a unique group of. Now we want to see what happened to the children. Okay? At 10 years, we visited again 135 of these families, and when the kids were about 12 or 13, we imaged the brain of these children, 92 children, and I'll just show you some of the finding. They're like a wealth of data, and I just want to show you finding related to synchrony, to oxytocin, and to the brain basis of attachment in the children. So here is what we're going to see: 9-month synchrony, at 6-year oxytocin, and at 12 years the brain basis of attachment. First thing I'd like to show you, just so that you get a feel, is the interaction at birth. What does it mean? How does it look? Okay, so here is what you see. This is a healthy mom, and you see the expression of those nonverbal behavior, the gaze, the vocalization, and you see that the mother is changing the frequency and intensity of her vocalization with the newborn. It's a 2-day-old baby with the newborn's very scant attention span. But what I really like is the first sign of synchrony in human life. At the end of this maybe 60-second video, the infant picks up his hand. This is a reflex. The infant, it's a newborn. The infant doesn't mean anything. Mother takes the infant hands and does this, makes it into a social signal. So this is the first sign in a healthy human's life where mother takes the child reflex and turns it into a social dialogue. And in comparison, you'll see the interaction of the depressed mom. Affectionate touch, the human licking and grooming. And she's looking at him, and you see the touch becomes, when he's attentive, and it's matching to the infant's rhythms. The infant is holding the gaze as much as he's able to. Here is our synchrony. See? So the infant becomes part of the diet. Now let's see the other one. Just a selection. We put them in the hospital in a room, and we ask them to interact with the infant for a couple of minutes as they wish. We don't give any instruction. The video is on. No, she doesn't. No, we film it from. We put the camera and we go out, so she's not in the presence of anyone. So you see that this kid is not really getting the emotional diet that he needs. Mother puts the child, and we see it all the time with depressed mom. Mothers put the child in a condition that will not enable the face-to-face intimacy. And there's very little expression of maternal behavior. So the infant doesn't even have the building block from which he can build a social dialogue. What happened in 9 months is that what we see often is that the mother of those that interrupt the intimacy. So depressed mothers, when the infant looks at them, they will tend within the next 2 seconds to turn their gaze as to signal to the child that this is too much for them. They're not able to hold the gaze. And the same when the infant positive vocalization, the mother will gaze away. So imagine a 9-month-old infant. He begins to vocalize to the mother, and within 2 seconds, the mothers will tend to gaze avert. And when you do transitional probability, we look at how it looks. We begin with moments of gaze synchrony. A moment of gaze synchrony is when I signal to someone else that we're on. We are in an interaction, okay? That takes for a depressed mother 5 times longer than it takes for a normal mother. Okay, so we have a moment of gaze synchrony. The first to break the synchrony is the mother. The mother breaks her gaze. This leads to one of those 3 outcomes. The infant breaks the gaze, or gaze averts after the mother breaks the gaze. The infant stops vocalizing, and the infant reduces positive affect. And they cycle within this for 7 times longer than, because interruption of synchrony happens, those cycle happens in a normative mother-infant interaction. There's a gaze aversion and resuming, gaze aversion and resuming. But it happens in a certain pace. And over here, it takes very long to trigger the first moment of we are together. Mothers break it very quickly, and it takes very, very long for this dyad to repair and engage in synchrony again. What are the consequences of this? At 6 years, those children from low-risk middle-class home, 60% of those children receive a psychiatric diagnosis by the time they enter school. So imagine if depression would come on top of all the other conditions, like poverty, like disrupted environment, like war-related trauma, et cetera, et cetera, et cetera. And the disorders are mainly anxiety disorders and conduct oppositional disorders, and this falls along gender line. Girls would tend to be more anxious, and boys would tend to be more conduct. It's only in the transition to adolescence that we begin to see that anxiety and conduct turn into depression. By the time children are 6, we don't see depression yet, but by the time they are adolescents, we see the cross-generation transmission of depression, vulnerability to depression. And what about the oxytocin system? We see much lower level of oxytocin in the mother, in the child, but also in the father. And this is strange because none of our father is clinically depressed. And here there are 2 explanations. Either the biological synchrony between mother and father, they tune each other neurobiological system, or something like birds of a feather stick together. There's some reason why these men, probably related to their own psychopathology, chose to marry those chronically depressed women. And this I'd like to show you some finding from the MEG. MEG is it's a brain imaging tool, less well known as compared to fMRI. Just in Israel, before a child is 17, you're not allowed to do fMRI on them. It's very different from this. So we used MEG to image the child brain. And what we did was the same paradigm as we did for the parents. We went to the home, we filmed the child interacting with the mother. A couple of years later, we showed this picture to the child, this video to the child, in comparison with an unfamiliar dyad. And by now you know what we would expect. That seeing your own interaction, seeing the attachment target, stimuli of the attachment relationship, will increase activity in the brain basis of attachment. And this is what we see. By the way, MEG, it has very, very good temporal resolution, so you could see the different oscillation, but it doesn't penetrate inside the brain. So you can only see areas that are on the cortex or right below it, but you can't see the amygdala or the subcortical structures. But here you see the same insula and STS, a very wide cluster of beta and gamma oscillations in response to own interaction. But what you see here in the children of depressed moms, particularly children of depressed moms who developed an affective disorder themselves, is not only there is much lower activation of these areas, but there is no differentiation between attachment and not attachment stimuli. So this is what living with a depressed mother does. Not only it depresses the activation of the affiliative brain, it limits the child's ability to fall in love, to enjoy close relationship, and then impair the parenting of the next generation. But it also blurs the differentiation between those we love and strangers. So children of depressed moms, because of not having experienced synchrony in infancy and not having developed a functional oxytocin system, it impairs their ability to respond differently to attachment targets than to strangers. And here we see that the activations are related to both synchrony experienced in infancy and to oxytocin levels, so that you see that synchrony experienced in the first 9 months of life and oxytocin that we collected at 6 years both impacted the degree of activation in the child's brain basis of attachment at the transition to adolescence. So we see a lifetime effect, and we see that synchrony during the first months of life plays a very important role. So I want to very, very quickly end by taking you to what we've been doing for the past couple of years since we realized how important it is to experience synchrony, and that it has to be experienced in the first 9 months of life. There's no time to wait. We need to act when the window is still open. We developed a dyadic intervention for postpartum depression that's based on the neurobiology of love, the system that I've just detailed for you. We're using social network. We found that this is the best way, and we have so many mothers contact us. We give 10 treatments at the same time, and we still can't account for everything we have. We started it in the Tel Aviv area, so we have mothers from all. And a few weeks ago, we got contacted from a hospital in the Galilee that serves the Jewish and Arab population. So now we have Arab therapists giving same treatment for depression to the Arab population. So this is our sign and mothers contact, and we have, this is basically what we do. Once we identify that the mother is eligible and we diagnose that she has clinical depression, we go to the home and we do clinical measures, behavioral measures, and physiological measures. We collect hormones from hair, from saliva, from breast milk, if there is breast milk, from nails, from the infant nails, and we also measure the microbiome to see for potential gut-brain interaction, either as causal factor for depression or as improving following the intervention. There is 8-week dyadic treatment trial. It consists of 1.5-hour session that involve a video feedback. Each session has a relational topic, like what is synchrony, what is intrusiveness, the importance of touch, emotion regulation, the notion of rupture and repair, so that mothers know that this is the way it should be, that interactions have ruptures and then they are repaired, and how you repair, the idea of mentalization and how to understand that infants have mind and that we can understand their mind and talk about their mind, express mindful things to them. And this is within an RCT, and it's compared to 8 weeks of developmental psychoeducational intervention where we go to the home. We still video the children, but we tell mothers about developmental milestones, so we don't work so much on the relationship. In our dyadic intervention, we film the mother at each session. We take the videos to the lab, we really look for them closely, and we mark moments of synchrony when the infant was looking for the mother, because one of the things that depressed mothers really have very strong is that they don't feel that the infant needs them. They don't feel the infant wants them, and a lot of time they say that the infant is better off without them. So we look for a moment when the child really wanted his mother, and we mark it and we help mothers. What we often see is that in the first session, mothers are very stressed and very intrusive, and they do a lot of things, but not in a face-to-face position. They're not able to be present, so they act, okay? Not being, but doing. At about session 3, mothers begin to engage in a face-to-face interaction, and about session 8, when we're successful, mothers are able to just be present and allow the infant to experience what Winnicott calls the capacity to be alone in the presence of someone, so that the infant can be with the mother and the mother doesn't feel like she has to constantly stimulate. And I have a video, very brief video from first session and last session from a mother. So I'll detail to you what you hear. She's making a lot of noise with a bunny. She's presenting the child with a toy, and it's a very noisy video. I'm sorry you can't hear it, but she's not looking at. There's constant stimulation. And there's no gaze synchrony. The mother and child don't look at each other. Okay, here you see she's singing to the baby, she's touching the baby, she's looking at the baby, and the baby's laughing. See, she's even getting closer. Very uncommon for depressed mothers, and they have a very nice toy interaction, a very nice face-to-face interaction. So you can imagine it, even if that's a peta cake, lots of touch. To conclude, bonding is a process through which the physiological and social signals of one partner synchronize with those of the attachment partners. The oxytocin system plays an important role in this process. Like mammals, humans can impact the physiological systems of the attachment partner through social contact and the experience of biobehavioral synchrony. Conditions associated with disruptions to social bonding: maternal postpartum depression, 18% of women, premature birth, approximately 12%, and environmental stress, I would say millions and millions of children each year, involve dysfunction to the child's oxytocin and maturation of the social brain. Because the oxytocin system is open to environmental influences, early intervention focused on touch and synchrony may help parents and children reorganize the neurobiology of attachment during the sensitive period for social growth. And I'd like to and just with a few words on the conceptual implication of our model, just like a very brief foray into philosophy. Gerald Edelman, who is, in my opinion, one of the greatest neuroscientists of recent decade, talks about the subjectivity gap. And he says that there is a huge difference between what we study, the brain's machinery, okay, what we do with fMRI or EEG or AEG, when we measure the activity of the brain's machinery and the feeling, emotions, and thoughts, mental life of the first person, of the feeling individuals. And that the gap, this subjectivity gap between what we know about the brain and the experiencing mind is not a gap that could be overcome by more sophisticated machines or greater precision or more money. This is an inherent problem that to examine the first person, to examine the mental life, it's insufficient to examine the working of the brain. Now, when we come to complex other systems, such as the solar system, or even the gut microbiota, maybe an objective description of how the system works is sufficient. Or when we speak about human life and how to make a difference in human life, just an objective description of the brain and the neuroendocrine systems is not sufficient inherently. And this is something that Husserl phenomenology claimed over 100 years ago in his paper on philosophy as a rigorous science, that there is no way that just understanding the objective operation of the brain would let us understand the mind. Now, what I am suggesting that perhaps we do not know for sure if our brain experience of reality is real or not real. But what we do know for sure is that we synchronized with another person. So while our perception of reality cannot be fully validated, our co-experience, our synchronous experience with someone else are not to be refuted. So perhaps this four-pole matrix of biobehavioral synchrony, how biology and behavior and synchronous experience are shared between two humans, can perhaps open the gate, define the language, or chart a new terrain to what Edelman called a brain-based epistemology, a science that can contain both our objective understanding of the brain and our subjective understanding of the mind. Thank you. So there's one study, I can actually, because I have preliminary results from a real study. So we have a study of teenagers, 14-year-old, and we do them in the hyperscanning, what you saw with the infants, and they are with their mothers in 3 types of interaction. Face-to-face interaction, Skype interaction, when mother and child are going to different rooms, they could see each other, and but they are not in physical proximity to each other, and screens, when they WhatsApp to each other. So when they WhatsApp, the whole story of brain-to-brain synchrony just goes. So whatever is needed to sustain that sense of being together from a biological perspective, and in the Skype, it doesn't go all together because we're still able to somehow synchronize. It's not as good. But when you go into technology that takes away the physical presence of someone, and we still don't know exactly what this physical presence. So we think, or one avenue we're going into is pheromones. So we're checking for oxytocin in sweat, because we think that this is partly what we communicate with. It's a very primitive mechanism for communication. So there's something about being physically present with someone, because obviously in the Skype, you can see that person's face and you can synchronize. They look at each other, they laugh together, they take turns, they match each other's speech inflections. So that reduces the synchrony by something. But when there is no gaze and there's just communication through the WhatsApp, there is no synchrony between their brains. So I hope I've convinced you that biobehavioral synchrony is a key experience of humans throughout life, both in the relationship with their mother and with the relationship with other attachment partners. So yes, I don't know how much of screen time is sufficient, you can do without it being disruptive. But if you don't keep that communication that is not through screen, there's something that maybe particularly in periods of reorganization for the brain, such as the transition to adolescence, there's all these hormones of puberty reorganize the brain. So during these times or other times when the brain reorganizes, the first 3 years, the 5 to 7, the transition to adolescent, those times you must maintain some face-to-face contact with real physical presence. The systems we spoke about are very environment dependent. That means that they're shaped by the environment, and that gives them one very important quality is plasticity. Those are very, very, very plastic systems. The affiliative brain is very plastic, okay? Let's say motor development is not very plastic, it's canalized, okay? Visual system is not very plastic, but those systems are very plastic and they can be repaired. So humans can repair bad early relationship by later benevolent relationship. This is the whole notion of psychotherapy, and we know love triumphs, and some people come from very bad beginning, and when they experience a stable and committed love, these systems could be repaired. Now, the question that we don't know for sure experimentally is the reparation full or the reparation is partial? Okay, so when there is early deprivation, there's got to be something to hold the reparation possible. I'd like to give you one example of this, which I feel to me is the most stunning example. This is an example from Albert Camus. You probably all heard about him. There's a beautiful book that was published 35 years after he died. It's called The First Man. I think this is the English. I read it in Hebrew. I think it's called in English The First Man. And he describes a very impoverished early environment in occupied Algeria, aggression, poverty, abuse, unsafe neighborhood. He was born, his father died before his birth, his mother was deaf mute. Okay, so he really grew up without anything. However, he describes with very, very eloquent words the touched base synchrony that he had with his mother. It was this kind of physical closeness and touch with his mother. But that wouldn't make a man win the Nobel Prize for Literature. At the age of 13, his grandfather wanted to take him out of school and put him to work, and there was a teacher who took him under his wing and was an attachment figure to him and introduced him to the world of letters. He had a big library and he taught him. And so obviously, the brain of a genius is not the brain of everybody else. So what we see from the story, and it's a beautiful book, it describes what I said in a much, much nicer way. But it describes, that at the transition to adolescence, which is a period of transition, a new development of the brain, if there is a new love, someone who's considerate, somebody who teaches you, somebody who takes you under the wing and developed an attachment relationship, there is a possibility for reparation. But it also tells us that there must have been something in early infancy without which I would assume reparation would not have been possible. It could be a grandmother. It could be a nanny. It could be an intervention like Lily does or like we do. It could be something small, but something just to keep the fire on until the brain meets better investment. So I think these two things, and we know that the systems are plastic, but we don't know the extent of it. So I think there's much more work that needs to be done.
Again, help me thank Ruth for being here today.